Hello, everyone. Can everyone hear me all right? Okay, cool. And we are live. Thanks for tuning in, taking the time to today's live stream. We have the Peptide team here. We'll be talking about the agent itself, what we are going to do, and what's coming up in the pipeline. But before diving in, just some background on the current ignition sale. The Peptide ignition sale started last Friday, May 1st, and within minutes of the sale starting, it was already oversubscribed, which is amazing to see. And as we speak, the latest numbers we have is, let me check. We have currently 13.9x oversubscriptions and over 690K USDC committed by over 800 participants. So super, super amazing to see. Sale still continues. Sale ends Thursday, May 14th at 1 p.m. UTC. So it's going to be next week. There is still time to participate in the sale. So you can still go check it out on AppBio XYZ. And just as a side note as well, staking bio is one of the best ways right now to secure an allocation in the sale, as 20% of the total token supply will be airdropped to bio stakers at TGE. So go check it out. There's still time until May 14. And we'll also share more information on this one on our socials and in Discord. So keep an eye out. And with that, just to kick things off, I'm going to hand it over to Ella from Molecule Labs.
Hi, thank you so much, Edda. And nice to be on this call, everyone. It's always so funny doing a live stream because there are audience out there existing. But we've got a chat if you guys want to comment. We're always open to answer questions. But yeah, just a massive thank you to everyone for joining. Obviously, as Edda was saying, the Peptide sale took off last week. But I think it's kind of an indicator of a larger movement that's been happening across biotech and across medicine over the past couple of months to years, which is really this sort of incoming new peptide drug development, which kind of, in my opinion, kicked off around sort of the Ozempic era and the GLP-1s. And it really started this look from biotech into what peptides could do for health and how they might offer new sort of drug development candidates. And so sort of coming off the back of this renewed look into peptides, the Molecule Bio teams were essentially trying to figure out how we could use the tech that we're developing to apply it to peptides drug development. And so that's kind of where this project was born. Here with us today, we have Rafa, who is a scientist and sort of approaches things from the biology side of things. And then we have Martin, who is our AI tech head. And the two of them sort of work quite closely on Peptide, this Peptide project to sort of develop the systems and benchmarks and checks that need to occur. So yeah, and then of course, Molecule is here. We have our product Molecule Labs, which allows everything to be tracked and verifiable. So we are a supporting protocol for the Peptide project. So yeah, Rafa, do you want to maybe talk to everyone a little bit about the scientific process that is behind these fleet of agents?
Yeah, 100%. So maybe just to start on why peptides, it's because really peptides sit in some sort of like a sweet spot where they're more specific than small molecules. They're sometimes cheaper to synthesize, they're less immunogenic, and basically we can also work with non-canonical amino acids, which are non-natural forms of amino acids. So you can engineer them for stability, for selectivity, for half-life properties that sometimes natural amino acids don't have. And as you mentioned, Ella, the field is exploding right now. There's GLP-1 agonists like semaglutide, which are the best drugs in the drug class in the world right now. For peptide, we're starting with 3 lead targets. We start with the OX2R. It's also called the orexin receptor 2, which is implicated in sleep disorders, ADHD, narcolepsy. Basically, we already, with Bioprotocol, published a computational feasibility assessment for a first candidate, an 18 residue peptide agonist. And we also have an improved version called the OX2R-S1 that increases the stability and also the pharmacokinetics. So a starting proof of concept for peptide to put peptide on the map. We also work with the KIS1R receptor or kispeptin receptor, which controls the reproductive hormone axis. It's involved in fertility. And they're one of the first ones that will be sent to the wet lab, and it's an underserved area clinically. So there's limited treatment options here. That's also why we picked it. And we also have the GLP-1 receptor. So we're not trying to properly beat semaglutide head to head, but we're using it to prove that pipeline works because our autonomous agent can generate competitive GLP-1 candidates, and it works on any GPCR, which is a type of receptor that is specifically targeted for diseases. So now that I gave a little bit of a science meet, I want to go into how the feed works and how peptide.xyz, which is our website, looks like, and for users to take a look. So let me share my screen really quickly. So this is the website. Here we have the programs live, as I mentioned. There's 3. And then what we have is that we need around 8 gates to synthesis. The 9th gate is the wet lab. So if you scroll down, you can see that we have the fertility, ADHD. You can pick here, and you can see the candidates that we have. All the candidates go through the gates. So the 9 gates is the G1 gate is also called the target structure. So basically, do we have a high quality receptor that we can work with? Then we have the G2, or also called the conformational sampling. So basically, does the peptide adopt a stable 3D shape in solution? Then we have G3, which is for binding pose. Does it actually dock in the target pocket with high confidence? Then we test also for binding affinity, meaning what's the predicted binding energy versus known references. That's also a gate that requires wet lab experiments to be able to modulate it properly. G5, it's one of the, if not the most important. Basically, it's molecular dynamic stability. And it means, does the complex, meaning the receptor and the peptide, does it hold together over 100 nanoseconds of simulation? This is what we use. Then we have also proteolytic stability and ADMET properties. So will it survive in the body long enough to work? Is it toxic? Then we have also physicochemical properties. Basically, is it soluble, non-aggregating? And then we have a selectivity and safety. So does it hit the right target and not the wrong ones? This is also specific for having off-target effects. And then we have the wet lab, which is basically here. So the first, it's a binding assay that we do with Adaptive Bio. And this helps informs later stages whether we improve some of the candidates or not in later stages. So when we scroll down, we can see here what the agent thinks and how it's basically passing candidates through the gates. For example, if you entered the website a few days ago, you might have noticed that we had 10 candidates running on the KIS1R and fertility, and it dropped to 4. The reason is because we're working on a new candidate library. So the candidate library 1, the candidate 1, 2, and 10 passed with, or the candidate 8 is now passing the gates with successfully. And then we designed a new candidate that it's now in gate 3, the C2, and you can see here how the agent goes through these processes. So for example, the C110 passed, and then the C2 library 2 also passed. This is a new library that we're designing. And then we also designed a new one called the C3 library. So every time there's new candidates, the ones that fail will drop, and the ones that are performing well, we will see them up in the basically leaderboard, so we can see how they're moving. We can also see here on the OX2R that there's basically the 004 and the S1 variant as well. There's a few caveats, of course, but this is also something that we're also comfortable working with, because you don't need to pass all the gates in order to go to the wet lab. There is a go, no go at the end, that it's basically a human that decides whether this candidate should pass or not, depending on how the properties are looking like. So it's not a binary decision. It's something that we have the opportunity, for instance, if a candidate drops at G3 or G4, for example, to redesign it using generative AI tools. And basically we generate a new library, and they all pass through the gates before moving to the wet lab. For GLP-1, we have a few candidates also going through the pipeline. So you will be able to see how more candidates get added. Some of them, they drop, some of others get redesigned, and basically all the log is here. So by moving here, for example, the G3, it's submitted, then there's a new library design, then the candidate 10 passed gate 4. So subsequently, you will be able to continue visualizing this and see how the agent thinks. Here, it's another way of visualizing what it's working on, more direct. For example, the agent 1, which is the agent that is involved in GLP-1. Here is the gate, and this is the number of the candidate. We're also working constantly to be able to share this information in a way that it's approachable and in layman's terms, so that it's not so sometimes cryptic. But here it's a way to show that everything moves in the pipeline. There's awaiting decisions, or there's actually like running through different modeling. For example, gate 5, which is molecular dynamics, is something that takes around 2-3 hours, depending on the candidate to run. So it's something that it takes time. Sometimes it gets skewed because the agent is able to autonomously take decisions, but when it knows its own limits, it also asks for a human involvement. So I think we want to build something where we have the human in the loop, and then we're able to get our top candidates here on the leaderboard. We have the candidate C1 for C101 for the KIS1R receptor, and also they're both approved, and both for OX2R are also approved. So now we have 4 candidates that are going the wet lab soon, and we're constantly redesigning new libraries to see if they can improve upon those. Yeah, so this is a little bit the feed, and maybe I can also share some of the vision that we have, because at the end of the day, what we want to have is a fully auditable process where we can have open data here. And I think the most value comes from things like non-canonical amino acids, how they affect the peptide conformation. And now we're closing the loop with the first wet lab experiments, and then the agent is able to reason and to propose new experiments based on these results. We're also adding a layer as well for generative AI using tools that are from open source, like BoltzGen for those who are interested, or RF diffusion to generate better candidates. And also we have a screening, ADMET screening that is done for each of the candidates before they reach the wet lab, before gate 9. So yeah, maybe I will now leave the microphone or the space for Martin, who can tell us a little bit about more the architecture and how it works. And we can then continue with some questions.
Perfect. Thanks, Rafa. Yes, so I'll share my screen to have a bit of a backdrop for then picking up from where Rafa left off. So my portion, what I want to do is talk a little bit about behind the scenes, how does the agent operate? A high level note on that is that, to the spirit of DeSci, what we're trying to do here is, alongside the science, also then be able to share learnings on democratizing access to these tools and methodologies as well, to kind of expand how many people can participate in running these types of agents and being inspired by that. So this is a starting point to that process. And so to start things off, I'll start at the website, as Rafa was referencing that already, and I'll go down here to the gates just to speak to that side of things first. So really what we wanted to do was, having noted a few of the recent developments on the AI side, we wanted to find a way where we could coherently have an agent that can move through these gates and be able to do so using the best possible AI science tools at any point in time, having optionality around that as new things develop. And then importantly, also being able to play with the level of autonomy. So one thing I want to note just up front is that I think it's worthwhile to almost frame this similarly to how people talk about autonomy when driving, where you can have different levels of autonomy relative to how much the human is involved. So that might be an interesting kind of framing. And I think what we're doing with this journey is learning what is the right balance there, so that you can achieve more and more scale, essentially, but still ensure kind of the quality control through that process. So that's been a great journey so far, and something that we want to report on as we continue, just to be able to kind of set a let's say a baseline with a clear kind of true goal as an outcome, where it is taking these candidates through the quality gates and through to the wet lab. So moving over to then the more kind of visual view of what's happening behind the scenes with these gates. Essentially what I wanted to highlight here were a few of the different pieces that we're looking to bring together, and then I'll move on to the behind the scenes of how the agent is structured to do so. So essentially the quality gates give us this kind of repeatable process that the agents travel through. And as they travel through them, at the bottom here, they're using different scientific tools, AI tools that are available. Currently, as you'll see here, the bio team developed a great tool in BioS that is used in the early phases to help source the candidates and so on. And then we've been working with a great team at Lightfold to use computational science tools that they've made available to walk through the follow-on gates. And then, as Rafa mentioned, for the wet lab, it's then where Adaptive Bio have built an API-based approach where we can then, the agent can essentially take its findings and find the top candidates and hand that off to the wet lab in a programmatic environment. And alongside using those science tools, it'll also be reporting what it's doing to Molecule Labs, which is then the on-chain lab where it can keep track of developments. I'll get back to that in a moment. And then for those of you who have been watching closely, you might have seen the experiment that was Beach Science. Now, just as a bit of a preview statement there's a V2 of this in the works, which will speak to part of the community engagement. I'll leave that for a later conversation, but that's just to say that there will be new, let's say, capabilities for how the agent can speak to the community that are being worked on there that we'll be aiming to integrate over time. And so to achieve all this, I think what was very exciting in the AI space, for many of you who have been following, I think in November, December last year, there was really a kind of, let's say, new level of capability that was demonstrated, notably with the continued takeoff of tools like Claude Code. But then also, towards the latter half of last year, you saw OpenClaw and its, stratospheric, let's say, rise to prominence. And so when it comes to the approach that we've taken, what was really exciting there, and what I think this OpenClaw concept showed, was that this new layer on top of a lot of these agentic tools had emerged, which presented itself as a very interesting opportunity, specifically when applied within the scientific research context. And so I want to take a step back just to briefly explain that high level, to then dive into how we apply it. And Andre Carpathy, a while back, tweeted about this kind of new claw harness. Now, OpenClaw kind of, I guess, pioneered this, but you've seen now things like Moose Research's Hermes agent and Anthropic themselves incorporating a lot of OpenClaw style capabilities. And I think that just goes to show kind of how powerful a UX this new layer is providing. And core to how we're approaching things on the PeptAI side is that it, fundamentally what I think it gives you is really optionality. And what I mean by that is that, by having this claw harness, you choose where it's going to be hosted. You can choose what models it's using. You can choose what skills it's using. And very importantly, it can have channels through to where, the different, for example, human experts that you want to reach out to are used to communicating. And so that's where, when you take all of that together, it becomes a very powerful new vehicle for this type of work. Just as a concrete example, with the agent fleet that we have today, Rafa gives a lot of his input via Telegram, which is then a channel that he's very familiar with. And I think these are kind of, let's say, the cumulative UX pieces that unlock a new way that you can engage with a broader audience, where, a lot of the, for example, experts that you want involved in this type of process will typically not necessarily have the time to learn lots of new interfaces. So the fact that this can basically be put into their existing workflows, and it has, a proven track record of how that's been achieved, is a big unlock for enabling what we're trying to do here. Also then, on this concept of autonomy, with the heartbeat approach, which is a very simple concept but a very powerful one, where it can be set to regularly check in on how are things going. You can then imbue it with increasing levels of complexity of a choice of, okay, I got the results back. Can I make a decision independently of whether to move ahead or not? And this is one of the key levers we've been working on, is like, depending on the results that come back through the computational analysis that's done in the gates, should it take a decision to move ahead by itself, or should it check in with a human expert? And as we move forward, we'll be able to essentially continuously review where the sweet spot sits, of course, with the goal of scaling how much an agent can do independently, but with that expert in the loop at critical points. And so that's just to speak to kind of why this claw harness is such an enabler. And of course, there's a huge ecosystem around these, as many of you will likely be aware, which means that the open source community around that means that we can, really compound on the learnings that are being shared across the work that's being done there. And we want to contribute back to that as well. So more specifically, when it comes to then what this agent looks like in our context, so the way that it works is essentially, I won't go into too many of the details here. I'll assume some knowledge of the claw harness, but just to hit the high points. With the agent harness, essentially what it gives us is a way to A, choose what models it's operating with, but B, with the tiered memory, this is where then the gates sit. So basically we imbue it with a kind of the best practices of this gate methodology that Rafa has outlined. And this is what it's referencing as it goes through. Now, what's really interesting, and I'll speak to this in a moment as well, is that as you have more agents, they will also be finding new findings that may lead to them updating, their understanding of these gates. And what we can then do is actually monitor across all the agents, where could there be shared learnings and so on. So I'll give a quick input into this in a moment, but that is a huge enabler in this context. Then, of course, as many are probably aware, there's the skills element of this. So importantly, what we're trying to do is keep it to the fewest amount of skills necessary at any point in time to be effective, so that we can kind of get a sense of what is working and what isn't, and then add complexity as we need it. But for the moment, then, what we're using for today is that the agents have a Privy agentic wallet. I'll come back to that in a moment in terms of how it interacts with X402 compatible tools. But then also it has skills for using the different scientific tools that it requires at the moment. And so alongside Privy, we have Aura and Molecule Lab skills for interacting with Molecule, the Molecule Labs and Molecule ecosystem. We have a BioS skill for interacting with BioS, and then the Lightfold skill and the adaptive skill will be used for the wet lab portion. And then, as I teased earlier, there's a new beach science that will be coming out that it can also have. So this is really how each agent, like from a high level, can operate. And I think a big unlock, and what's very exciting from the on-chain element of this, is that what we've already seen is that we're then kind of pushing the frontier of what's possible in terms of using, for example, X402 to make payments, especially in the science space, where what's great is we're engaging then directly with these partners and putting X402 as a priority on their roadmaps as well. So that's something to mention as well, is that this is very much a exercise of where we are pushing the envelope of what's possible, and then inviting more adoption of it, because as has now been shown by what we're very excited by the enthusiasm around the PeptAI launch, because this shows that we can now pair this programmatic funding with the programmatic use of that funding to make use of these tools, and that is a very exciting pipeline to start connecting all the dots of. And then be able to have a landing page that shows progress, because fundamentally this is about removing the bottlenecks to progress in a responsible fashion. And I think we're really getting all those pieces are starting to sing, to put it that way, in a choir together. So that's very exciting to see take shape. And as we do that, we invite more tool makers into that ecosystem, because there will be an incentive, of course, to become X402 compatible when there are agents knocking on your door saying, hey, we'd like to use our funds to make use of a great tool that's been built. Please make it X402 compatible. And that's just to say, I think that's an exciting aspect of this relative to the broader on-chain ecosystem. And I imagine we'll get to some, if there are any questions, please put them in chat. We can get to them after I've spoken. So that's a little bit on then the agent loadout side of things. And then just to speak to kind of a higher level view, kind of a little bit more behind the scenes that I mentioned, is that this means that we can basically use tools, for example, like Claude Code, to basically monitor across the agents. And this gives us a way to kind of scale the human expertise on this side as well, whereby we can understand, a human having to check as we scale up more agents, it would take a lot of effort to do that manually. So of course, you want to have systems behind the scenes to also be able to monitor what's happening and understand where are there shared learnings that could be deployed across them. And to this point of level of autonomy, you both then have it for the individual agents, but you also have it on the meta level of managing all of the agents. And we can increasingly then, this is really kind of the next step now that we've seen, we have the candidates ready to go to the wet lab with PeptAI so far, will be the self-improvement cycles that we can now put in place for the agents, and how they can learn from each other while having human oversight of all of that as well, so that we can stay grounded in knowing that these are substantive progress that's being made, all of it going back to getting more candidates successfully through to the wet lab and seeing good results coming back there. So this is how we're operating at the moment behind the scenes to really scale this in a way that gives us full transparency as we go. So what I wanted to speak to just as an example from the website is that alongside then seeing the gates here and the overview, we then the agents have already then used X402 to set up their Molecule labs, which is very cool, because Molecule, being a pioneer in this space, are X402 compatible for setting up labs. And this is where it will then document kind of its work. And one thing I wanted to note is we'll be working on the cadence of how to balance what you can see, on the site with kind of live information and what should be documented in the lab, because every gate, publishing every gate might be, it might create, so much of a pipeline that it might be overwhelming for someone seeing it. So right now we might start by, for example, our goal will be on Friday to capture a lot of what Rafa mentioned, for example, with this new library that's coming out, the reasoning behind that from the agent, and have it publish an announcement there. Possibly on a weekly cadence to start with. But this is also where we'd love community feedback, like, maybe there's an appetite to see more frequent updates. And I think Ella will speak to this in a moment, but the great thing is that this gives us that starting point of having an on-chain verifiable audit trail of what the agent has done. And Molecule Labs also has this capability of private and public information. So once wet lab results come back, those can be held in a private state potentially, versus sharing some of the computational analysis publicly. These are variables that we can play with overall with what is best for moving the project forward successfully, for delivering on taking these candidates as far as possible and creating an exciting outcome from that. So that's just to say that this is where we then, we want to play with this modularity so that we can create multiple views on the information. You can dive deeper if you want to. You will see the verifiability, and it, as we move forward, we can, double down on what we see works, and also update anything that, where the community might wish for greater transparency. We can really work towards that with these tools that we have available. And one other example, just to mention on that, kind of jumping back to this piece, is that we're also very excited because, there's new capabilities coming out, at a very high pace now, with people exploring new models that can be used, that could be relevant to these different gates. And the great thing with these agents is that we can then, for example, fork them and test a new pipeline with new tools and see, did it improve against a baseline? And this becomes not trivial, but we've built out the infrastructure to make this possible to do very rapidly, so that we can now move into this, self-learning phase that could then, create models based on the data that it's getting back, look at how can it plug into, DeSci primitives that have been built over the times with new types of community input and so on. I think, there's a lot of exciting work that's gone into building tooling that these agents then are hungry to make use of and test, but always tying it back to these high-level optimizing towards these outcomes that then can be verified in the wet lab. So that's really diving through that side of things. And then another element that I think is worth highlighting is, Paul, the co-founder and across both bio and molecule and the kind of, steering the ship in many ways across these two initiatives. One of the exciting pieces there is that he's posted about some of the work already. And I just wanted to highlight that, this got 734,000 views. And I think what's really exciting about this portion is to say that it opens up for a whole new paradigm of how you can aggregate feedback to what is happening. And we invite both positive and critical feedback, I'd like to say, because that allows us to then feed that back to the agent. And beauty with these agents is they have no ego. So if you critique them very harshly, that is great, because that allows us to stay true to saying, okay, thank you for that. We can incorporate it. So this is what we want to try to do. If this work is put out into the world and there's people who then give feedback on like, this is a terrible idea, this is a good idea, that can be incorporated into this entire pipeline. And because of the agentic nature, we can fork, we can update, we can move very quickly at that scale. And I think that's very exciting, because a lot of this, traditionally was siloed, was very small communities. And this gives us a way to invite a lot of eyes across this work and incorporate that learning as we go in a transparent way. And then coming back to the Mall Lab, we can also then, look to how can we even say, we've been monitoring across feedback and that influenced these announcements. This is all the variables that we want to play with going forward. And then always with a view to how do we improve, the outcomes with the agents that we have available. So those are some of the pieces that I wanted to introduce today. And as I mentioned, very happy to both get feedback, but also answering questions. And I really see this as an opportunity to have the technology that allows us to meet the scale of both the speed of scientific AI, but also scale up the community feedback aspect of it and be able to use programmatic funding to really enable all of that. So I'm very excited to see the progress and the excitement around the launch of the ignition sale. So yeah, love to hear back from the community. And with that, I imagine I will hand it back to Ella.
Yes, you've done such a good job of explaining everything. I always find that you have a perfect balance of technical and also grounded explanation. So thank you so much for that, Martin. And you also did a really great sort of intro of how Molecule and Molecule Labs fits in. But yeah, for those of you who don't know, Molecule has been in the DeSci ecosystem for, I think maybe like 6 years now. Sort of started bio spun out of Molecule, and we've always been really focused around building sort of the IP protocol sort of pipeline from proof of invention to IP NFT to intellectual property tokens. I saw they were mentioned even in one of the questions, and that it has all been great. But after sort of being in the DeSci ecosystem for a while, we saw a much bigger need, to Martin's point, of ways to track and trace the projects that were spinning out of the DeSci ecosystem. And so that was where our product, Molecule Labs, was kind of born. And we're super excited to sort of help this peptide project and have them use it. They're actually sort of our trial run of an agentic use of Molecule Labs and what that might look like in a sort of agentic world. And I think for me, jeepers, sorry, my light just fell off my table. But for me, I think there is, of course, always, like when you work on being on the bleeding edge of science and the bleeding edge of technology, which is what DeSci is. It's kind of really throwing all of technology's capabilities at science and seeing what clicks, what works. And that is really exciting, but there can also be a stage where people don't really know like what's hype driven, what's legit, and I understand how tricky that can be, which is why I think like meeting the team, like having calls like this where you get to meet Martin and Rafa and the sort of brains behind these projects. I think that's a really helpful like proof point. And then of course wet lab validation, which for those of you who don't know, wet lab, basically you get a wet lab and a dry lab, and dry lab is all computational stuff. And a wet lab is basically taking it into like a real laboratory and testing it in like actual organisms or cells or mediums and that sort of thing. So that's kind of, and biology is, she's a nasty beast, in that sometimes you can predict and computationally model, and then something you put it in a cell and it just doesn't go how you plan it, which is why I think this wet lab validation is very important. But anyhow, all of this is to say is that data then gets tracked in molecule labs where everyone can come and verify it, validate it, and see it, and track the progress of how these agents were thinking, the decisions they made. And I think, obviously I'm biased, but I think that's quite a critical step in developing these agents, in having a community-led understanding where people contribute to these sort of larger projects, and sort of co-develop drugs together. I mean, honestly, like we're looking to build a community around these projects, and sort of have a greater body of people work on them, and that purely happens through adequate and proper communication where people can connect and see and track and sort of follow a mission. So that is our hope for how Molecule Labs obviously assists in this project. We're very, very excited to sort of have it run out. And I think it's also nice for us to have a room to experiment, you know, sort of Martin and Rafa will shoot out to us and be like, hey, we need this. What would you think of this? And then our product team sort of gets on it. And so I'm very excited for the next couple of months. Obviously, the audience is super excited too. We've seen that the Ignition sale has been crazily oversubscribed, and it's, you know, still got many days to go. So I do feel like we've got a bit of there's a spark and an energy here that I think the moment feels right for. And so, yeah, very excited to work on that. And then maybe if we want to do some of the questions.
Cool. Let's check the chat. Okay, the first question is, who makes the human decision on whether or not a candidate passes to the wet lab? And is this something that the community of peptide holders has governance over, or how does that work? I think we've briefly mentioned this. I think, Martin, you briefly mentioned this, but yeah, if you want to maybe tell a bit more about it, it would be very helpful.
Yeah, so I think so for the moment, essentially the way that this works is linked to this kind of levels of autonomy piece. So right now for the first agents, essentially that's where Rafa, with his scientific let's say background, is key in making some of those calls. And so that is the approach for the moment. But now I think this gives us then the baseline, noting what Ella mentioned around the experimentation. Once we have these go through to the wet lab and have completed this first cycle, it then becomes something we can review now for what is the optimal level of autonomy to take to the next level, basically. Should there still be this kind of go, no-go call linked to one expert, or is that something that could benefit for new runs in a more community scenario? So I think that's something that will be, that for this first version, it will be focusing on, for the ones that you see on the site currently. Rafa is really sitting in the key role for making that call, but it gives us then the reference point for how much additional autonomy to get, because that's also then, in terms of use of resources, you know, a bigger decision to be made. But it's a lever that we can essentially pull on based on the learnings. On the science side, Rafa, I guess maybe if you want to add some your thoughts from that perspective, but on the technical side, that's the, let's say, starting point.
Yeah, just nothing to add. I think there's really the question where the gates that we've built together really provide a very good platform to be able to decide whether something is worth advancing to the lab or not. There's also a question whether like these results are going, there's no bad data basically. It's all good data fitting into the system. The system is self-learning. So at this point where we have identified our targets and our receptors, it's just a matter of building the data layer so that there can be better predictions.
Cool. Thank you. And actually, also, we have some more questions from Nootropics DAO on the proof of invention protocol and on-chain minting of IP, how this relates to the development of these candidate molecules.
Yeah, so I can jump in here. The agents have actually been minting their own IP NFTs, which you can see in Martin shared one of the links. So yeah, they're able to autonomously interact with the molecule protocol and use it in that way. But kind of, I think what I was sort of trying to get to earlier, which is that I think obviously our IP protocol about intellectual property tokens and IP NFTs, they are kind of the rooted core of the molecule protocol, but with Molecule Labs, we're looking to extend things a lot deeper. So sort of looking at access management, encryption, and that sort of thing. So we're trying to offer a bunch of services essentially to these agents. But yes, the agents are out there minting IP NFTs, doing their thing, all also alongside the X402 work.
Thank you. And the next one is, again, from Nootropics DAO, around the architecture of the agent. Each receptor target has a given target. Is that correct? Can you elaborate on the role of these as an economic actor beyond X402 payments? How are they originally funded with experimental budgets? Are they tokenized, or do they accrue any of the commercial utility that results from developments in the experiments that they fund?
Rafa, would you like to take that one, or?
Yeah, we can also divide it in two. I think
Yeah.
on the
Yeah.
on the X402, you can take that part. So yes, there's an agent for a receptor right now. There's multiple architectural, let's say, ways in which we're thinking, maybe aggregating and iterating on those ones. But for now, each receptor has an agent, and the agent is able to iterate based on that. There's learnings that are made, and therefore the agent has the most important thing, memory and context, to be able to take decisions and to pass candidates through the gates. And there's right now the sale that we are finalizing, or is going to be finalized in the next 9 days. It's going to leave a 25K budget out of the 50K that are initially raised. Those are going to be all for wet lab experiments. Right now we have given around 10K for the KISS1R receptor, around 7.5K for OX2R, and 5K for the GLP-1s. This is also subject to change, but this might stay as it is. And basically, this is to have the first round of experiments, and then take a decision whether we need a new round of iteration in the candidates. And this is also when we open the channels basically for the community to come with their own receptors or their own targets. Because I believe there's another question also asking something around, can I or when can I basically ask for new targets? So this is in the next step. We're going to be unlocking funds after this 25K, depending on how the data comes. And then we can continue with other receptors as well and doubling down on what we have. But maybe, Martin, you want to, before the next question, also say something around the economy on the X402?
Yeah, so I think you answered most of the question because it was kind of beyond the X402 piece. But I guess just to dovetail on what Rafa said, exactly as he mentioned, some budget has then been allocated to these agents at this point. And one thing to note is that on the Molecule Labs, there is an item for experiment cost where this initial budget is also listed for the two agents that have a Molecule Lab already. And it's just to say that then from there, that's where then the X402 can be used to make use of that budget where the tools that it uses accept it. But I think you probably covered the going beyond that.
Cool. Can anyone run an experiment?
Not now, not on the first iteration, but on the second one, we'll be open to new targets. So if you have new targets that you believe are interesting or where there's an underserved medical area, please keep us in touch. Once we have the first results, we'll open this for the community, and this is where we can collaborate together and we can have your targets run, or even your candidates if you want to validate them through our stack. If they're interesting, we can also discuss that.
Thanks, Rafa. What does a successful launch look like for the Peptide or BioVision in 6 or 12 months from now? What different milestones or timelines would you look and say, this isn't working as well as I hoped it would, or wow, this exceeded my expectations?
I can also take part of this one. I think it's a great question, so thank you for that one. Well, I don't think there's like a way where this is not going right, in the sense of it's data at the end of the day is data. So we're publishing data, and this is like the valuable, the most valuable part of it. In general, this is also something that we're thinking about constantly, is what are the assets that can be generated through the pipeline. If it goes the best way, lead candidates can be and assets, peptides basically, can be generated, and then we want to take them up to pre-IND when we have binding assays and CAMP, so basically cell-based assays as well, making sure that there's no off-target effects. And this is usually when pharma can come and we can license them out, or they can like just straight up buy the assets. We have seen this over the last 2 years, has happened in small biotech companies and AI drug discovery companies as well, helping pharma reach out these stages. This would be let's say the best case scenario. And then that's why we also build programmatic funding within our architecture, is because we want to go step by step. So first we unlock a certain part of the funding. We do our first round of experiments, and then we decide, do we change direction? Do we double down? And then do we open this for community, for new targets if the results are positive? So it's quite modular in that sense. But yeah, let's say worst case scenario, we have a very valuable data set on non-canonical amino acids, on peptides and toxicity properties, and basically on prediction power, whether this new generation of the generative AI works or not. So it's all valuable. Martin, do you have anything else?
No, I would say my fundamental thing is very much like you mentioned at the very beginning of your answer. I think for me, it's all leading from the data and the science, essentially. Like seeing, I think what's very exciting about this is that we have a pathway to having a programmatically funded journey for AI agents to substantively make progress on taking candidates through to the wet lab and generating that data. And then the possible outcomes of that, as you described. So for me, it's really already now getting this phase underway and getting that first data back will be very exciting. And then over a 6-month period, best case is that that flywheel really takes off, essentially. So I would say, to the degree that we're able to facilitate that, that's really the journey that we want to take, but being very led by, I think there was another question that kind of spoke to, what is the decision on whether this, it was the same question, whether this isn't working or not. That will be very much informed by what we see in the results from the data. So I think that's, my overall approach is setting up this scaffolding so that we can make data-driven decisions, basically. But as you noted, what's great is that even if the data is negative, there are scenarios where that could be interesting data, essentially, for improving models and so on.
Amazing. Is there a token tied to Peptide? I can take this one. So currently the Peptide Ignition sale is on BIOS Launchpad. It started last Friday, May 1st, and it's still running for another 9 days. It concludes May 14th. So next week, there's still time to participate in the sale. And after the launch ends, there will be TGE. So you can learn more about it on app Bio XYZ, about the launch itself and the current status. We'll also share the links in the chat if you want to take a look at it. So yes, there will be a token tied to Peptide. We have a follow-up question from Nootropics DAO. One of the ideas behind the agents minting the IP NFTs for the candidate peptides, that the agent keeps part of the initial supply in its own custody and then actually hold further sales themselves to fund subsequent stages of the candidate's pipeline to extend their respective budgets and become at least partially self-funding.
I can give a short answer to that. Let me know, Rafa, if you want to dive in. This is very much one where what the right way to answer would be to see. I think you're. It's a very good question, and it let's say looks in a direction that I think, you know, if phase one goes in a promising way, it's one of the levers that becomes possible to pull, to put it that way. So but leading with the data that we get as a result, this is exactly the types of options that become available by leveraging things like leveraging molecule labs, essentially. So I think that's what's very exciting, is that we then can do this phase one, get the data back, and then make an informed decision of like, what is the best path forward now? But you know, the tools available from molecule labs enable exactly that. So, but that's one of the options, to put it that way. So I wouldn't say it's something that is like committed to from phase one, but it's something that becomes possible as a result of it.
Cool. How do you decide the funding allocation?
Rafa,
Yeah. So the funding allocation was decided depending on what we saw on the preliminary results. So we identified these 3 receptors, and then where we found the most promising candidates were on the KIS1R receptor, also as an underserved area. So that's why there's a bit more funding that goes there, because there's several candidates that look very promising. And the more data that we have, the better predictions that we make. So in this specific case, that's why we allocated a bit more for KIS1R. This is the one that is the most advanced in the pipeline. But that doesn't mean that there's not enough funding going for the rest. We planned enough to have experiments so that we get enough data for the candidates in general.
And maybe just to dovetail on that, that's an example of where for phase one, to this theme of levels of autonomy, once we get that data in, it gives us more signal to then decide on how we can optimize that further, essentially, going forward, and what is the optimal funding allocation mechanism relative to the data that we then gather for scenarios where we can, you know, what can we automate further, but very much grounded in real data.
Amazing. Thank you. And I think the last question, if we haven't missed anything else, focused on human trial outcomes, possible applications in augmenting and scaling organoid production.
Not like really up our alley, as we're focusing on the first step of the drug development process. But later on, yes, I mean, if we can test these peptides on organoids directly, like ideally at some point there's a human trial outcome, but it's not really what we're thinking right now. It would come in later stages. But yeah, hopefully.
Nice. If there are no other questions, maybe we can wrap things up here. And if there are any more questions, feel free to just ask them in our Discord channel. Yeah, we'll be also sharing more updates on the current Ignition sale and also on Peptide. Follow us on X at Peptide. where you can see all the details of the roadmap, what's coming up, what's currently being worked on. And yeah, with that, you can find also more about PeptAI on our Launchpad. I shared it in the chat. You can go there, check the roadmap, and also still participate in the sale. And yeah, thank you so much, everyone, for tuning in. Yeah, see you soon.
Thanks,
See you soon.
everyone.
Thank you.
Bye.
Bye.